Hey there, science enthusiasts! Ever wondered how we cracked the genetic code? It all started in the 1860s with a friar named Gregor Mendel, working in a humble garden in what's now the Czech Republic. But Mendel wasn't just gardening for fun, he had a mission. He crossbred pea plants, focusing on 7 traits like height and flower color. Mendel's goal? To understand how these traits were passed to offspring. After years of meticulous work, he discovered some groundbreaking principles. First, he figured out that traits are governed by distinct units of inheritance, what we now call genes. Each plant had 2 copies of a gene for each trait, 1 from each parent. These copies could be identical or different, and he called these variants alleles. Mendel also noted that one allele could dominate the other. If both were present, the dominant one masked the recessive. He then found that each gamete, or reproductive cell, contained only 1 copy of a gene for each trait. When a male and female gamete combined, their alleles determined the plant's traits. Next, he discovered that alleles separated during gamete formation. This is Mendel's law of segregation. And finally, he deduced that the segregation of one pair of alleles didn't affect another pair. This became his law of independent assortment. Sadly, Mendel's work went unnoticed at the time. It wasn't until 1900, 16 years after his death, that 3 botanists independently rediscovered his findings, cementing his legacy in genetics. So next time you look at a pea plant, remember, it's not just a plant, it's a pioneer of genetics. Thanks for watching and stay curious.